So it's not surprising that I start with this slide where I introduce what it's an emerging concept of how a genetic disease may actually have an epigenetic pathogenesis. And this signaling through dystrophin activated nitric oxide nitrosylation of HDAC2 in this case, and disruption of the inhibition of HDAC on MyoD is actually the first example of the epigenetic regulation of gene expression in muscle cells due to the loss of one component of the dystrophin-associated complex. So as HDAC and MyoD control the coding and decoding epigenome, and in particular muscle genes and a myogenic microRNA that are commonly defined as myomeres, it's very important to step back and go to what we have learned in the last 10 years on the control of muscle gene expression at the chromatin level. So before differentiation occurs, muscle genes are normally repressed by a number of additional, redundant, and collaborative mechanisms that include the deacetylation performed by two different classes of histone deacetylases, class 1, especially HDAC 1 and 2, and class 2, especially HDAC 4 and 5. And then deposition of marks of histone marks of repression, such as the methylation of lysine 9 of histone 3 and the trimethylation of lysine 27 of histone 3, which is mediated by the polycomb complex. However, these inhibitory constraints of muscle gene expression are removed and replaced by activatory signals as soon as the cells are exposed to differentiation cues. So in this case, we can imagine differentiation cues as all those signals which are emanated from the regeneration environment. As you can see, upon the activation of several signaling pathways, all the repressors are replaced by a number of activators, which consistently with the idea that there is a transcriptional epigenetic switch, depose a number of completely different epigenetic marks that are coincident with the activation. So in the absence of histone deacetylases, the acetyltransferases PCAF and P300 induce the local hyperacetylation, which relaxes the chromatin. And then there are a number of other components which induce arginine and lysine methylation. But most importantly, at least to this talk, is the recruitment of this Y-SNF chromatin remodeling complex, which is endowed with two alternative subunits, ATPase, BRG1 and BRAMA, which use the ATP energy to remodel the chromatin and allow and displace the chromatin from the DNA and then allow the transcription. So the Y-SNF complex is an extremely interesting and evolved one. So if you look at the composition of this complex from yeast to mammals, you will certainly observe, that a number of components is increased, and more than this, some of these components are duplicated, if not triplicated. For instance, you can see here that in addition to BRG1 and BRAMA, the two catalytic subunits, there are two components of BAF53, which is a structural components. All of them, except for BRG1 and BRAMA, are all structural components, whose function is still the object of debate. Most of them have been proposed as interacting protein that make contact between SWI/SNF and the transcription factors where the SWI/SNF should be recruited. And BAF45 has 3 variants. BAF60 has 3 variants, and we will focus a lot of this seminar on these components. And the general idea is that these are lineage-specific variants which may confer on the SWI/SNF a sort of functional specialization. So depending on the composition of this complex, the complex can be used by different cell types for different tasks. So to start to elucidate the function of BAF60, we have adopted a complex approach consisting of the RNA-mediated knockdown of the 3 BAF variants. This is wrong, BAF60A, B, and C, and BRG1 as a control, a gene expression profile, a chip analysis, and the chip-seq, which is still running. But you can see already from here that the normal myotube formation is not disrupted by downregulating BAF60A and B, while it's clearly disrupted by the downregulation of BAF60C and BRG1. I will show you later on the gene expression profile. However, what was clear from the very beginning of the analysis is that knocking down each of these 3 components has a specific effect on completely different subset of genes. Knocking down BAF60A has an effect on fibroadipogenic genes, suggesting that BAF60A is important in the activation of the fibroadipogenic genes. The B was more specific for the adipogenic genes, and the C was specific for the muscle genes, indicating that the BAF60C-based SWI/SNF complex is the complex that helps MyoD in promoting muscle gene expression. So we started to analyze the interaction that BAF60C can make with different components of the myogenic transcriptosome. And we use MyoD, as MyoD is the initiator of myogenesis, BRG1 because it's the catalytic subunit of the SWI/SNF complex that promotes MyoD-dependent gene expression. And as you can see, the first surprise was that in GM, which means in myoblast, before induction of differentiation and muscle gene expression, there is already by co-immunoprecipitation the formation of a complex between BAF60C and MyoD in the absence of BRG1. While in myotubes, the whole complex composed by MyoD, BAF60C, and BRG1 was detectable. And by a proximity ligation assay, we could verify this interaction in vivo. As you can see, BAF60C and MyoD interact equally in myoblasts and in myotubes. But BRG1 and MyoD interaction occurs only in myotubes. BRG1 and BAF60C could weakly interact in myoblast, possibly away from MyoD, obviously. And this interaction is enriched in myotubes. So this first analysis suggests that BAF60C and MyoD can actually form a complex away from the BRG1-based SWI/SNF complex. And we wanted to verify whether this complex could be formed on the chromatin of MyoD target genes. It's important to emphasize at this point that the current view in the field is that the SWI/SNF complex with all the components travel always as a whole complex, while these data are now suggesting that certain components of the complex like BAF60C can make interaction with transcription factors like MyoD away from the whole complex. So when we did the chromatin immunoprecipitation on a typical MyoD target genes, myogenin, we observed that in myoblast, before the activation of myogenin, MyoD and BAF60C could be already detected on the chromatin of myogenin, but BRG1 and another constitutive components of the SWI/SNF complex, INI1 or BAF47, were not detected. and this was coincident with the relative hypoacetylated conformation of the promoter. However, during differentiation, the promoter was now occupied by MyoD, Bas60C, and the other components of the Swesny complex, BRG1 and INI1, suggesting that there is a two-step recruitment in which Bas60C MyoD interaction precedes the engagement of the other components of the Swesny complex. So we therefore decided to understand whether Bas60C has an active role in recruiting MyoD on target genes. So we knocked down Bas60C, and we made a chip-on-chip analysis on MyoD, known MyoD target genes. So those are all known MyoD target genes that have been described by Tapscott and by Int'l. So in all of them, in the absence of Bas60C, the MyoD binding was greatly impaired. As you can see, these are 4 examples, and the red line corresponds to the RNA interference of Bas60C, indicating that Bas60C is important for MyoD to recognize the target genes prior to the activation of gene expression. And consisting with the model in which before transcription in myoblast, there is a complex formed by MyoD, Bas60C, perhaps already P300, which somehow marks MyoD target genes or poise MyoD target genes for the activation of transcription as long as the core Y-SNF complex is recruited. So what are the signals that allows the recruitment of this Y-SNF complex during differentiation? And we know since a long time that a very strong signaling pathway that induce muscle gene expression in myoblasts is the P38 pathway. And in particular, among the 4 isoforms, alpha, beta, gamma, and delta, alpha and beta are extremely important, are necessary for the activation of the myogenic program. So we and other groups established that phosphorylation of MEF2C, the MyoD heterodimer partner E47, and coordinate the recruitment of this complex on muscle genes. We showed last year that phosphorylation of P38 removes the polycon complex, engage the polycon complex to repress PAC7. So in a way that P38 can activate muscle genes and repress those genes that are not needed for muscle differentiation anymore. But the problem was that we already knew that P38 kinases were important to recruit this Y-SNF complex. So it was kind of easy to hypothesize that the phosphorylation of some component of this Y-SNF complex by P38 alpha or beta could be the event that mediate the activation, the recruitment of Y-SNF complex and the activation of muscle gene expression. So we started to analyze the phosphorylation of many components. We focused on Bas60A, B, and C. And as you can see, we found a threonine, which is a highly conserved proline-directed threonine in mouse. Threonine 229 is highly phosphorylated by P38 alpha and beta, and this phosphorylation is abrogated when this threonine was replaced by non-phosphorylatable alanine. And the same information came out from the phosphopeptide analysis. And the importance of this phosphorylation is underscored by the fact that while the Bas60C wild type, after induction of differentiation, can interact with BRG1, a phosphorylation deficient Bas60C, in which the threonine 229 was replaced by alanine, was unable to enrich the interaction in differentiation conditions. So the current model we can envision is a two-step recruitment of the SWI/SNF complex. First, through the component, one component, BAS60C, which makes the interaction of MyoD. And upon phosphorylation of BAS60C, the recruitment of the BRG1-based SWI/SNF complex would allow the chromatin remodeling. But we wanted to test this new model of activation of muscle gene transcription in a system, in two complementary systems. The first one was the typical myogenic conversion imposed by MyoD on fibroblast. As you can see, normally, when fibroblasts expressing MyoD have a very poor myogenic conversion as long as they are kept in a growth medium, which is conditions that are not permissive for differentiation. However, if we induce the differentiation by activating the P38 pathway with the upstream kinase and KK6, as you can see, MyoD becomes able to induce muscle gene expression and myotube formation. And this is paralleled by the activation of myogenin. So when we did the chromatin immunoprecipitation in these conditions on myogenin promoter, in the absence of P38, we could observe BAS60C and MyoD on myogenin promoter, but not BRG1. And as soon as the P38 pathway was activated, we could find BAS60C, MyoD, and BRG1. Again, suggesting that P38-dependent recruitment of the SWI/SNF complex occurs by a two-step. And this can be even better studied if we take cells that do not express BAS60C. So we found that HeLa cells, which are notoriously resistant to MyoD-dependent conversion, lack the expression of BAS60C. So the first experiment was actually the one of reintroducing BAS60C in HeLa cells and realizing that upon introduction of BAS60C and activation of P38, HeLa cells could express myogenin in the presence of MyoD. But the second experiment was more definitive. We purified the SWI/SNF complex from HeLa cells. And this complex is deficient, is without BAS60C. And we decided to do an in vitro transcription assay where we use a chromatinized MCK muscle creatine kinase template where MyoD is supposed to activate transcription because there are two MyoD binding sites. And we incubated in vitro the all HeLa SWI/SNF complex, which is deficient of BAS60C, the MyoD 47 force dimer, and then the kinase, P38, the purified P38 alpha kinase, and the recombinant BAS60C. And as you can see, in the absence of BAS60C, you have to assume that MyoD is always present. So MyoD with the SWI/SNF core complex from HeLa does not activate transcription. If we include in the reaction BAS60C, wild type or mutant, no activation of transcription, unless we introduce the P38 kinase, which phosphorylates BAS60C, and this is coincident with the activation of transcription. While the phosphorylation-resistant BAS60C mutant was not able to support transcription from the SWI/SNF and MyoD-activated template. So the overall model is now that in the absence of differentiation cues, myoblasts sort of expose an epigenetic mark just to let the SWI/SNF complex know where to reach the MyoD target genes. So only in cells with MyoD, BAS60C is recruited to this promoter in a seemingly silent conformation that, however, signals where the SWI/SNF complex should be recruited upon the phosphorylation. So this is the two-step signal that somehow provide a completely new or novel view of how this Y-SNP complex is redistributed. We want to propose that the chromatin redistribution of this Y-SNP complex is dictated by the components making interaction with different transcription factors and additional signals inducing this Y-SNP core complex, which is the one enzymatically active, reaching this interaction by specific signals. What is the connection between this basic knowledge and muscular dystrophy or regeneration in muscular dystrophy? As I mentioned in the first slide, there is an HDAC supervised network that is very important to activate transcription, muscle gene transcription, and therefore regeneration in dystrophic muscles. And we have found this interesting network by which HDAC controls an interplay between myogenic microRNA and Bas60C variants. And this came from an initial observation. I will take a step back, that the use of HDAC inhibitors, which are FDA contain actually a number of FDA approved drugs, when applied to a mouse model of muscular dystrophy, were able to induce regeneration, but also reduce fibrosis and adipose infiltration. And we could never find a molecular link between these two activities. However, if you remember, Bas60A and Bas60B are important components of the transcriptional machinery that promotes the expression of fibrogenic and adipogenic genes. What are the fibrogenic and adipogenic cells in the context of skeletal muscles? These cells. They were described a year ago by two different groups as they were called FAPs, fibroadipogenic progenitors, by the group of Fabio Rossi. And these cells in normal muscles are normally not myogenic, but can facilitate myogenesis indirectly by cell-to-cell interaction with the satellite cells, which are the professional muscle stem cells. However, they display an adipogenic in response to injury. When these cells can be easily sorted because they are SCA1 high, SCA1 positive, and then they share with satellite cells CD34 as a surface marker, and they are negative for CD31 and for alpha-7 integrin, which is instead a marker that distinguishes satellite cells. So when we isolated these cells from dystrophic muscles, we initially found a sort of surprise. Normally, in muscles from normal mice, these cells are extremely boring. They look like fibroblasts. You can culture them for 20, 30 days, and they look like fibroblasts. And if you treat the mice with TSA, you cannot observe much changes. However, once isolated from MDX mice at the early stage of the disease, when the MDX mice are highly regenerative, because during the disease at the beginning, the dystrophic muscles tend to regenerate continuously. Then we found that these cells, especially when exposed to trichostatin A, a deacetylase inhibitors, started to form something that could really look like myotubes. So when we analyzed the gene expression in these cells by immunofluorescence, we found that these cells, taken from dystrophic mice treated with TSA, could actually make muscle in vitro, as you can see from the expression of myosin heavy chain in these multinucleated cells. And most importantly, this was coincident with the reduction of the fibroadipogenic potential, indicating that these cells can actually determine whether dystrophic muscles can undergo regeneration or fibroadipogenic degeneration. And for those of you guys who don't know, the pathology of muscular dystrophy, the fibroadipogenic degeneration is the most deleterious outcome of the disease. As you can see, in the presence of trichostatin A, a number of muscle genes were upregulated, and fibroadipogenic genes were downregulated. So we did a microarray of these cells in different conditions, and we confirmed that a number of genes upregulated by TSA were included in the muscle development, contraction, and lineage acquisition of these cells, while a number of genes involved in the adipogenesis were downregulated. And most interestingly, when these cells isolated from dystrophic muscles were compared to satellite cells, we found that in MDX treated with TSA, the number of genes that were corresponding to the genes that are usually expressed by satellite cells became close to the 100%. 80% of these muscle genes that are normally expressed in satellite cells are now expressed in muscle interstitial cells of MDX mice exposed to deacetylase inhibitors. And this is the list of the genes for. This network poses that after exposure to agents that inhibit histone deacetylases 1 and 2, a number of muscle genes are induced. This is because of the recruitment of the Zweig-Sniff complex. But we also know that, I mean, it's actually now 10 years after the first discovery, that in addition to muscle genes, there are microRNA that are typically expressed in muscle, like myomeres, whose promoter typically resemble the promoters of muscle genes. They have an inbox which binds MyoD, and we found that this by chromatin immunoprecipitation, these promoters enriched by MyoD and HDAC1 and 2 were the promoter of microRNA 1.2, 133A, and 206. So overall, by analyzing the muscle interstitial cells at different stages of disease progression in MDX mice, we noticed that these cells are highly plastic at the very beginning of the disease, consistent with a highly regenerative potential, and they form spontaneously fibroadipocytes. In this case, there are droplets of adipocytes, but when treated with the trichostatin A or any other deacetylase inhibitors, these cells tend to reduce the adipogenesis in favor of the myogenesis. However, as soon as the regeneration cues decline, the pluripotency of these cells decline too. And this correlates with the lack of response of all MDX mice to deacetylase inhibitors, to the point that we're trying to step to a clinical trial with deacetylase inhibitors, and it's now clear that only early stages of the disease are susceptible to the beneficial effect of these drugs. So this is clearly emphasized by the positive effect of trichostatin A in young MDX mice. As you can see, the cross-sectional area which reflects the induction of regeneration is induced in young dystrophic mice, but not in all dystrophic mice. The elimination of the adipogenics deposition is present in young but not in old mice, and the same for fibrosis. So what we thought is that simply transplanting muscle interstitial cells taken from young mice into old mice could restore the activity of deacetylase inhibitors. And this was actually what we observed. Just focus on the cross-sectional area, and you see that in old mice that were injected with muscle interstitial cells, trichostatin A is now able to induce an increase in the cross-sectional area, which reflects their contribution to muscle regeneration. So I guess my time is over, and I just want to thank the people that contributed to this study, in particular Sonia Albini, Sonia Forcale, who has now a lab in Barcelona, Lorenzo Chiara. Chiara now moved to her postdoc in France. Valentina Saccone and Silvia Consalvi were still actively working on this project in Rome. So I thank you for the attention, and I'm ready to take any questions.